Welcome to the deep dive. Today we're tackling a really a complex puzzle. How our genes and maybe things affecting our genes link to a serious lung condition. Pulmonary arterial hypertension or PAH.
Yeah, it's a tough one. Our mission for this deep dive is really to unpack some fascinating research looking at these chemical tweaks to our DNA, things called DNA methylation.
And how they might play a role in PAH.
Exactly. We're focusing particularly on one gene, TET2, and, what happens when it's maybe not doing its job properly.
Right. We're diving into a study from 2025 by Heinmarch and colleagues. The title is a bit of a mouthful, but it tells you a lot. TET methylcytosine dioxygenase 2, TET2 mutation, drives a global hypermethylation signature in patients with pulmonary arterial hypertension, PAH.
Correlation with altered gene expression relevant to a common T-cell phenotype. Phew.
Yeah. Basically, they looked at DNA methylation patterns in blood, comparing healthy folks to people with PAH.
And crucially, comparing PAH patients with TET2 mutations to those without. Then they tried to link those methylation patterns to gene activity data.
Okay, let's get started. For anyone maybe less familiar, what exactly is PAH? Why is getting down to this molecular level so important?
Well, PAH, pulmonary arterial hypertension, it's a really severe disease. The blood vessels in the lungs basically remodel themselves.
Remodel? How so?
They get thicker, stiffer, narrower. Imagine trying to push water through a narrowing pipe. The pressure builds up.
Right, that makes sense. Increased pressure.
And that leads to all sorts of problems. More inflammation. The immune system gets involved in unhelpful ways. We often see changes in specific immune cells, particularly T cells.
T cells, okay.
And also more scarring, fibrosis in the lung tissue. Even changes in how the cells themselves generate energy, their metabolism. It's complex.
So that's the what, but what about the why? The study mentions genetics, but also epigenetics. How do those fit together here?
That's a key question. We know about, over 18 genes that, if mutated, can lead to inherited forms of PAH.
Okay, so there's definitely a genetic component.
There is. But here's the thing. Mutations in those known genes, they only explain some of the cases, maybe around 20% if the cause isn't obvious, what we call idiopathic PAH.
Only 20%.
Yeah, and maybe just over half, like 54%, in families where it runs, familial PAH. So for a lot of people with PAH, those known gene mutations aren't the full story.
Which means something else must be going on.
Exactly, and that's where epigenetics comes in. It's like a layer of control on top of the genes.
Epigenetics. That's the stuff that changes how genes work without changing the actual DNA code itself, right?
Precisely. Think of it like software instructions for your genetic hardware. Epigenetic mechanisms can change how active a gene is.
Like turning the volume up or down.
Kind of. DNA methylation is a big one. It involves adding these tiny chemical tags, methyl groups, onto the DNA, often at specific spots called CpG sites.
Okay, methyl groups, and what do they do?
Generally speaking, when these tags get added, especially near the start of a gene, they often reduce the gene's activity. They sort of tell the cell, ignore this gene for now.
So they act like off switches, or maybe dimmer switches.
Dimmer switch is a great analogy. And then you have enzymes, like the one made by the TET2 gene, whose job it is to remove these methyl tags.
So TET2 helps turn genes back on, or keeps them from being turned off too much.
Exactly. It helps maintain that balance, which is why this study zoomed in on TE2.
What was the specific thinking there? Why TT2 in relation to PAH?
Well, TT2 makes this demethylating enzyme, right? The one that removes the tags. And some earlier work had suggested that mutations in TT2 might actually be a novel cause of PAH.
A new cause? How common?
Pretty rare, based on initial findings, maybe in less than 0.5% of PAH patients they looked at. But it was a clue.
A clue suggesting what?
Suggesting that if TET2 isn't working properly because of a mutation, maybe those methyl tags aren't getting removed as they should.
So you'd end up with more methylation.
That was the hypothesis. That PAH patients with these TET2 mutations would show a kind of global increase in DNA methylation across their genome. The dimmer switches would be stuck in the down position more often.
Okay, interesting hypothesis. How did they test it? What was the setup?
They compared blood samples from 3 groups, 10 healthy controls as a baseline.
Right.
Then 10 PAH patients who didn't have mutations in any of the known PAH genes, and importantly, they also checked they didn't have TET2 mutations.
Got it. PAH, but no known genetic cause.
And the third group, 9 PAH patients who specifically did have TET2 mutations, but again, no mutations in the other established PAH genes.
So they were trying to isolate the effect of TET2?
Pretty much. Then they used a technique called reduced representation by sulfite sequencing, RRBS.
RBS, what does that do?
It's a way to map DNA methylation across the genome. It doesn't look at every single possible spot, but it focuses on regions that are rich in those CPG sites where methylation typically happens. Gives you a really good representative picture.
Like checking key intersections instead of every single street for traffic.
That's a good way to put it. After getting these methylation maps, they compared the patterns between the groups.
Yeah.
And then, crucially, they took those findings and compared them to gene expression data, basically, how active different genes were.
But that gene expression data wasn't from the same patient.
No, that's an important point. It was from a separate publicly available data set of other PAH patients. So it's a correlation, not direct proof in the same individuals.
Okay, correlation. So what did they find when they compared the methylation maps?
Okay, finding number one. PAH patients, in general, showed more DNA methylation than the healthy controls.
More methylation overall.
Yes, about a 0.776% increase in the PAH group without other mutations, and a bit more, 1.124% increase in the group with TET2 mutations.
So higher in both PAH groups compared to healthy, but even higher with TT2 issues.
Exactly. But here's a really interesting detail. In the PAH patients without TET2 mutations, the extra methylation seemed to be concentrated on specific chromosomes, certain areas. But in the patients with TET2 mutations, they saw significant hypermethylation, extra methylation, across all chromosomes. A pan-chromosomal effect, they called it.
Wow. So TET2 mutation affects the whole genome's methylation level, not just specific spots.
It certainly looks that way based on this study. It suggests TET2s plays a really broad role in keeping methylation in check across the entire genome, and when it's faulty, you see this global increase.
That really highlights the impact of TT2. Was that the second key finding then, that TET2 mutations made it worse?
Yep, that's essentially key finding too. The PAH patients with TT2 mutations didn't just have more methylation than controls, they had significantly more than the other PAH patients without the TT2 mutations.
Which fits the idea that TET2 removes these tags, right? If it's broken, the tags build up more.
Precisely. It's consistent with TT2 losing its demethylating function.
Okay, so we've established there's more methylation, especially with TT2 mutations. But what does this actually mean for the person, for the disease? Does it affect how genes work?
Right, that's the crucial next step. Finding 3 starts to connect those dots. The researchers looked for specific regions where methylation levels were significantly different between the groups. These are called differentially methylated regions, or DMRs.
DMRs, hot spots of change.
You could say that. They identified these DMRs and then cross-referenced them with that public gene expression data set from other PAH patients.
And did they find a link?
They did. They found that many of the genes located in or near these hypermethylated DMRs in their study group actually showed lower levels of RNA, meaning less gene activity in that external data set of PAH patients.
So the increased methylation seems to correlate with decreased gene expression. The dimmer switch is turned down and the light output is lower.
Exactly. It strongly suggests a functional consequence. The methylation isn't just sitting there, it seems to be actively suppressing these genes.
And which genes were being suppressed? Did they look at the functions? The title mentioned T cells.
Yes. This leads us to key finding 4, which is where it gets really interesting biologically. They performed functional analysis on the genes that were both hypermethylated and downregulated.
What did that analysis show?
It showed a really strong enrichment for genes involved in, you guessed it, T cell functions. Things like T cell differentiation, T cell activity, terms like lymphocyte and T cell kept popping up.
And this was true for PAH patients generally, or just the TT2 group?
Both, actually. This pattern was seen when comparing controls versus all PAH patients, and it was even stronger when looking specifically at the differences driven by the TT2 mutations.
So T cells seem central here, maybe affected differently depending on TT2 status.
That's what it looks like. They dug deeper into that. They identified over 1,000 DMRs that seemed specifically linked to the TET2 mutations.
1,000 regions, wow.
Yeah, and analyzing the genes of those regions again highlighted T cell functions. They found 65 unique genes with this TET2 specific hypermethylation linked to 11 enriched T cell pathways.
And were those genes less active too?
Good question. Yes, 23 out of those 65 genes also showed reduced expression in the external data set. So it's a specific set of T cell-related genes seemingly being silenced due to hypermethylation linked to faulty TET2.
Okay, that's the TT2 specific effect. What about the general PAH effect on T cells, the one independent of TAT2?
Right. They looked at that too. They found 98 genes that were commonly hypermethylated in both PAH groups compared to controls, so regardless of TET2 status.
And did those point to T cells as well?
They did. Functional analysis of those 98 genes again showed enrichment for T cell terms, 24 different terms related to T cells, involving 15 unique genes from that list.
And expression changes.
Yep, 10 of those 15 genes also showed lower expression in the external dataset. So it really strengthens the idea that messing with T cell activity via methylation is a common feature in PAH, whether TT2 is mutated or not.
So methylation is hitting T cell genes from kind of two angles, a general PAH effect and a specific TET2 mutation effect. Were there any standout genes, ones that seem particularly important?
There were a couple they highlighted. One was EIF2AK4.
EIF2AK4. Does that ring a bell?
It should if you know about rare lung diseases. Mutations in EIF2AK4 are known to cause pulmonary veno-occlusive disease, which is actually a rare type of PAH.
Ah, okay, so it's already linked to the disease area.
Exactly. And this study found EIF2AK4 was hypermethylated and likely downregulated in their PAH cohort. Plus, other research has shown that reducing EIF2AK4 activity can mess with T cell populations.
So it connects the methylation finding back to both PAH and T cells. Interesting. What was the other one?
The other big one was TCF7. This gene makes a protein called TCF1.
TCF1, what does that do?
It's a really key regulator of how T cells develop and decide what type of T cell to become. Think of it as a traffic controller for T cell differentiation.
Okay, how so?
It generally promotes the development of less aggressive T cells, like Th2 cells and memory T cells, while putting the brakes on the more pro-inflammatory types, like Th1 and Th17 cells.
And Th1 and Th17 are often implicated in autoimmune issues and inflammation.
That's right. And those types are known to be more active in PAH. So guess what they found with TCF7?
Let me guess. Hypermethylated and downregulated in PAH.
Bingo. TCF7 showed hypermethylation, and its expression was lower in that external PAH dataset. This suggests a potential mechanism for the increased inflammation in PAH. If TCF7 is silenced, you lose that brake on the pro-inflammatory T cells.
And was this linked to TE2, or is it more general?
This was particularly interesting that TCF7 methylation and apparent downregulation happened regardless of TET2 mutation status. It was part of that common PAH signature.
So TCF7 could be a really important player in PAH inflammation across the board.
It seems plausible, yeah. It makes it a potentially very interesting target for future research or even therapies in PAH generally. Silencing TCF7 via methylation could be a key step in driving that inflammatory T cell profile.
This paints a really compelling, though complex picture. But you mentioned earlier the study had limitations. What should we keep in mind?
Definitely important to remember. First, sample size. PAH is rare, and TET2 mutations in PAH are even rarer. So the groups, especially the TET2 group, were quite small.
Meaning they might have missed some things, or some findings might not be as robust.
Potentially, yes. Small studies always have that caveat. Second, the gene expression data came from a completely different set of patients.
Right. The correlation point again.
Exactly. It strongly suggests a link between methylation and expression, but it's not direct proof within the same people. You'd ideally want both measurements from the same individuals.
Makes sense. Anything else?
They also noted a slight age difference between their methylation cohort and the public expression data set. The expression group was a bit younger on average.
Could age be a factor?
It could.
Right.
Especially since TET2 mutations can sometimes pop up with age in blood cells, something called CHIP, clonal hematopoiesis of indeterminate potential. But the researchers argued this was less likely to be a major confounder for the expression data because that group was generally younger, making age-related CHIP less prevalent. Still, it's something to be aware of.
Okay, so important context. Taking those limitations into account, what are the main takeaways, if you had to boil it down?
I'd say there are a few key things. One, PAH seems to involve a general increase in DNA methylation in blood cells.
Okay.
Two, if a patient has a TET2 mutation, this methylation increase is much more pronounced and spreads across the entire genome.
Got it. Panchromosomal hypermethylation.
Three, this extra methylation appears linked to decreased activity of certain genes.
The functional consequence.
Right. And four, many of these affected genes are involved in immune function, specifically T cell development and activity. This happens both in a general PAH way and a TET specific way.
And specific genes stood out?
Yes, finally. Genes like EIF2AK4, already linked to a PAH subtype, and TCF7, a key T-cell regulator, show this hypermethylation, potentially contributing directly to the disease features, especially the inflammation.
So what are the implications of all this? Where does this leave us?
Well, it strongly suggests that these epigenetic changes, these methylation patterns, are not just bystanders, but could be actively involved in driving PAH, maybe especially in those with TET2 mutations, but also more broadly.
Could this lead to new approaches?
Potentially, yes. The methylation patterns themselves, maybe detectable in blood tests, could one day serve as biomarkers, perhaps for earlier diagnosis or predicting disease course.
Biomarkers, okay. And treatments?
And perhaps new treatment targets. If we understand how methylation is silencing key genes like TCF7, or how TET2 dysfunction drives broader changes, maybe we can find ways to intervene in those processes. Targeting T-cell regulation or even the methylation machinery itself might become options down the line.
It's really fascinating work, connecting these tiny molecular tags to such a complex disease. It really leaves you thinking. Could we perhaps one day develop ways to specifically target and maybe even reverse some of these harmful DNA methylation changes? Could that offer a new way to treat or even prevent PAH? And what else is interacting here? What about environmental factors or other genes we haven't found yet? How might they play with TE2 and methylation to affect who gets PAH and how severe it becomes? Lots to think about still.
Absolutely. It's just the beginning, really.
Well, thank you for unpacking all of that with us today. A truly insightful deep dive.